Hello, everyone. Welcome back to the ML for Protein Engineering seminar. I'm excited today to host Nathaniel and Shri here, who are going to be, they're grad students from Duke, and they're going to be presenting their work on functional alignment of protein language models. And so I'll let them take it away from here, but join me in welcoming them.
Yeah, happy Tuesday, everyone. We're really excited to present our work on the functional alignment of protein language models via reinforcement learning. This was work led by Srinath and myself and Dr. Philip Romero's lab here at Duke with some amazing undergrads, Algrim, Sarah, and Amaya. And Jason, is it cool to answer questions at the end of the presentation? Like, feel free to leave them in the chat. Happy to
Yep,
go through anything y'all want to ask. Does that sound good?
yep, sounds good.
All right. So I would hypothesize that many of you have used ChatGPT, but you might be less aware how ChatGPT became the useful tool it is today. And so what OpenAI started with was this pre-trained generative transformer called GPT-3. This is a large language model that was pre-trained to predict the next token or word from text data. And so an example response is what we show here in the top row, where the model GPT-3 was asked to use the word serendipity in a sentence. And when you look at the response in blue, one thing you'll notice is that it doesn't actually use serendipity in a sentence beyond defining the word. And it also has some weird artifacts like repeating the phrase use the word in a sentence 3 times. And so while this response is syntactically and semantically valid, it's not a very helpful response to humans. And so what OpenAI did is after noticing this problem, they developed this alignment algorithm and they aligned GPT-3 and created this model called InstructGPT. This is the sister model to ChatGPT. And if you look at the blue response, this is the aligned model response, you'll see that it actually does a better job of defining serendipity, but also provides what was user intended with an example for this. When Lori first moved to the city and started her job search, it was serendipity that she stumbled upon the perfect opportunity and was able to start her career in marketing with a company she truly loves. And so you can see that this response is much more useful to a human. And that leads to the question of how did they achieve this enhanced performance? Well, they developed this algorithm called reinforcement learning from human feedback. And this aligns large language models to respond to prompts in a manner that more accurately matches human preferences. And so they start with a pre-trained large language model that predicts the next token or word. And there's this two-phase strategy. It begins a supervised fine-tuning, and this initializes a model to actually start taking the action you want, like responding to a question in a conversational manner. And then they do proximal policy optimization. But first, you have to do this reward model training. So this is training a supervised model on human preferences. And model is actually what guides alignment during proximal policy optimization. What they'll actually do is they'll sample from the large language model and then use the reward model to evaluate how well that response is human preferred or useful to a human. And when you look into our field with protein language models, I mean, there are many parallels because many protein language models are actually based on large language model architectures. But in our case, while they are like large language models, often transformer architectures, instead of being trained with text data, they're trained with natural protein sequence data. And what's really cool about these models is they've been shown to generate representations that capture how a protein's linear arrangement of amino acids encode its structure, function, and dynamics. And these representations have been powerful for a range of tasks from structure prediction, functional annotations, variant effect predictions, as well as evolutionary modeling. And we've noticed an uptick with protein language models also being used as generative sequence models. For example, this is ESM3. This is a state-of-the-art protein language model from the evolutionary scale modeling family of protein language models. And what was really cool about their preprint is they designed novel and functional green fluorescent protein variants that were very far in sequence identity from natural training examples. And this is really cool. You know, these models are designing functional variants far from these examples, which is kind of a great step towards de novo protein design. But when you look at these designs, as well as those from other protein language models, while when you look at the function of these, unfortunately, they're often not as good or beyond natural training sequence function. So essentially, these generated sequences are novel in sequence, but not in function. And this really leads to an issue with protein engineering objectives. So these models are misaligned with one of the core goals of protein engineering, and that's to create proteins beyond what evolution has explored. And when we believe this is because they are not pre-trained to gain an explicit understanding of protein function. And so if you look at what happened with the results of like GPT-3 aligned with RLHF to get InstructGPT and ChatGPT now and all these other models, it would be really awesome to have the same thing happen for protein language models and align these with human intent to generate proteins with biophysical or biochemical properties specified by humans. And so we introduce reinforcement learning from experimental feedback, or RLXF. This is a framework for aligning protein language models with experimentally derived notions of biomolecular function. And so RLXF is directly adapted from RLHF that was used to train ChatGPT, as we've been discussing. It begins with a pre-trained protein language model. In this case, we'll be focusing quite a bit on ESM2 models, and it will follow a two-phase strategy analogous to RLHF, where in supervised fine-tuning, we initialize the model to generate sequences in the correct region of sequence space. And then in proximal policy optimization, we then directly align sequence generation with the reward model. And before I go into a few more details about this, what's really important to this process is the reward model training. So at the core of RLXF is a user-defined reward model that modifies the base protein language model to generate sequences with high reward values. In principle, this can be any sequence-based or structure-based scoring function. It can be a machine learning model. It can be just an equation you have for solubility. It can be a zero-shot predictor. There are evolutionary POTS models. You can do Rosetta-based energy functions. You can even do multiple of these all at once, like a multi-objective criteria. In this work, we focus on supervised models trained on experimental sequence function data because they offer highly accurate predictions of complex protein properties. This is something our lab spends quite a bit of time on. And by learning from this experimental data directly, we found that simple neural networks guides are they're capable of modeling these protein properties even when the biophysical mechanisms are poorly understood. And we would like to give that knowledge to the pre-trained protein language model. And so going back to this two-phase process, and the supervised fine-tuning phase, we use the reward model to explore sequence space, and we identify a small, high-quality collection of high reward sequences. And then when you fine tune the protein language model on just this handful of sequences, it provides a strong bias to sample amino acids observed in these sequences with high reward. And in the PPO phase, the sequences are sampled directly from the protein language model. So for example, the blue model here is what to focus on, where we take a batch of masked CreiLOV sequences. We use aligned ESM2 to generate probabilities for amino acids at those masked positions. And then we sample from those amino acids. But then we use the reward model to actually predict the reward of these sequences. And over time, this reward that we calculate, uh, increases, and the likelihood of sampling amino acids that increase this predicted reward also increase. And I'm happy to go into as technical as you want to. So please feel free to drop some more technical questions in the chat regarding our implementation of SFT and PPO. I tend to find with most audience, it's great to go broad first and then go technical. As PPO is, it's to me, when I started this, it was very hard to get a handle on everything that was happening. Um, and so one of the things we did was evaluate the generality of RLXF. So we applied it to 5 diverse protein classes. They span a range of sequence links, structural folds, and molecular functions. So GB1, AVGFP, UBE4B, BGL3, and PAB1. And so for each of these systems, we trained a supervised reward model with deep mutational scanning data. And this was what was used to guide the alignment. And then on this row where we're showing these win rate plots, we evaluated the generative performance using a win rate metric. This is very common in natural language processing. What this means is it's the percentage of times a model generates a sequence with a higher predicted reward than a reference model. And so in this case, it's pre-trained ESM2 650 million. And so we are really excited to see that across all of these proteins, different protein classes, different molecular functions, the win rate significantly increased after SFT and PPO in most cases, except for this one case with ESM2 8 million. But one thing about this is we noticed no model size consistently outperformed others. The best performing model really varied on the specific protein system. Now, we did notice originally that the smaller models could be less stable during training, so we would probably recommend starting with ESM2 150 million or ESM2 650 million. But if you wanted the best results, I would look at the individual likelihoods of all the single mutations in the sequence and compare those to literature values, and you can kind of see how well you're aligned for your specific protein. And then to conclude here, like what you'll see with many of these plots is the largest gains often came from the SFT phase, with more modest improvements after PPO. But part of this is because SFT models had already achieved near perfect win rates, while in others, this plateauing may reflect our limited hyperparameter optimization. Um, so I'll go into more detail on what happened after SFT and PPO with the key protein that we actually characterized in vivo in a little bit. But one more figure about generalizing to these 5 protein classes. We are really excited, if you zoom in on the top model in each one of these cases, you can see that the distribution of predicted function. So in x-axis here, we have the predicted function of each one of these proteins, and on the y-axis, you have density. You can see that after SFT and PPO, you see these dramatic shifts in predicted scores. And what was really cool too is that when you look at not only generating 5 mutants like we did on the first row, on the second row, we generated up to 20 mutations for all these classes. And we saw that models aligned after SFT and PPO still were outperforming the base model, indicating that we could really go quite far in terms of how many mutations we could add and have a good chances of introducing mutations that increased the protein function. But we wanted to verify in vivo if these results held up in the wet lab. And so what we chose as a target to engineer is CreiLOV. CreiLOV is an oxygen-independent fluorescent reporter protein, and it's uniquely suited for use in hypoxic or anaerobic environments where the ubiquitous oxygen-dependent green fluorescent protein, or GFP, is non-functional. And this makes CreiLOV a promising tool for applications in low oxygen settings, such as gut microbiomes, tumor microenvironments, and high density fermentations. So it's really quite useful to humans if we could. But an issue is both natural and engineered variants of CreiLOV are substantially dimmer than GFP. So they're actually quite hard to use in the lab. Actually, Dr. Romero's wife, Dr. Venturelli, her lab has tried to use this protein and found it quite hard to use because it's just not very bright. And another thing to note about this is the love domain in CreiLOV that allows it to be fluorescent. It was actually evolved as a blue light photoreceptor, so fluorescence is not actually part of its biological function. So when you think about these models, like protein language models trained on natural sequences, they're not very likely to encode the information needed to enhance a non-natural property like this. Um, but there's a need for this. Like just like we see in many cases in protein engineering, we want to have generative sequence models that are aligned to generate sequences with the non-natural properties enhanced. And so we wanted to actually, verify if these pre-trained protein language models were actually misaligned to enhance non-natural fluorescence. So what we did is we predicted the fluorescence of 5 mutant designs from ESM2 protein language models, the more recent ESMC models, as well as a variational autoencoder. This is a generative sequence model that is trained on a multiple sequence alignment. And so it's trained on natural sequences related to CreiLOV. And the biggest thing we saw repeatedly was that ESM2 650 million actually predicted the greatest max and mean predicted fluorescence for designs. Um, and then when you look at this win rate plot, really interesting, we saw that model size also did not scale with win rate percent. So when you look at the ESM2 models, it did increase up to 650 million, but then dropped quite a bit with a 3 billion parameter ESM2 model. And we're still not sure why this is happening, but ESMC, we actually saw that as model size increased, you saw pretty dramatic drops in win rate percent. And you saw this reference model in this case was the pre-trained VAE, which did the worst typically. And this is why these results combined is why we focused on aligning ESM2 650 million. And when you look in a little bit more detail on the pre-trained ESM2 models, we found that they really did favor the natural CreiLOV sequence over mutations known to enhance fluorescence. So we actually went through the literature, went through the DMS data, and we pulled out key mutations that are known to enhance fluorescence. And so this heat map on the y-axis, you have amino acid identity. And then on the x-axis, you have sequence position of CreiLOV. And what we did is we masked each position of CreiLOV to get logits and then did softmax to get probabilities. And those are colored using this color map. And so you can see this C43, this is a rationally engineered substitution that disrupts the native photocycle and significantly increases fluorescence. And you can see pre-trained ASM2 basically gives it a zero probability of being added to the sequence. So what that means is anytime this position is masked, it's putting back in the native cysteine, which basically kills fluorescence. And then you see T7S. This is the best known or the brightest single variant discovered in the CreiLOV DMS dataset. And you can see that the wild type residue with these black circles is preferred as well as another, I think that's, but you basically T7S had a very likely chance of being added to the sequence, which just goes again to show that if you were to just take the pre-trained protein language model and see what its probabilities were for adding a mutation, it could very likely steer you in the wrong direction. And then we did RLXF. And so these results kind of show in silico what the improvements we saw in the model were. And so we saw the functional alignment with RLXF significantly improve sequence generation. Again, this is these predicted log fluorescence on the x-axis, density on the y-axis with CreiLOV as the reference predicted log fluorescence. And you can see a dramatic shift from pre-trained to SFT to PPO. And again, this was with 5 mutants, but we wanted to see how many mutations we could add before the generative performance dropped off. And we were really surprised that we could really add up to 45 mutations before they dropped below base model performance, indicating that these aligned models really could add quite a few mutations and still enhance fluorescence. And so one of the reasons why we see such a dramatic improvement in predicted fluorescence of designs is because the sampling has changed so much. And so here we're looking at how the functional alignment dramatically shifts sampling distribution towards landscape regions with high fluorescence. And so this is an MDS plot where we took the DMS training data, the wild type, and sequences generated from all the ESM2 models, pre-trained SFT and PPO, as well as the VAE and the aligned VAE. And we basically just did the Hamming distance of all of these. So it's just comparing the sequence identity. And so when you do the multi-dimensional scaling of this and do take the first two dimensions, what this means is that when sequences are closer in space, their circles are going to be closer on the plot. And when they're further in sequence identity, they're further away. And so what you see is the pre-trained VAE in pink and the pre-trained ESM2 model in light blue. They actually have quite a bit of overlap, which you might think because they're trained on natural sequences. And you can see that after alignment, um, you'll see after SFT alone, they're now anchoring in a different region of sequence space. And when you look at the same plot but overlaid and colored with predicted log fluorescence, you see that this space of SFT has dramatically improved predicted log fluorescence, and that's further refined with PPO. And so what this means is the sampling is much more efficient, and now you can only take a handful of sequences, and you're much more likely to have enhanced properties. And when you try to go and see like what key mutations it learned, we looked at known mutations again in literature for enhancing fluorescence. And you see, like I mentioned, A43C, it reverts back to the native cysteine that kills fluorescence. The pre-trained model does. But on the aligned model, you no longer see that. And you see like T7S, the best known single mutation variant, dramatically increases in the number of variants that it, it increases dramatically in how many variants from the model are generated have that. So.
Cool. Okay, so before we show you guys some of the key results that we're excited to share, I wanted to back up a little bit and just explain some of the optimizations that we made along the way to really optimize our model performance. And so the first sort of thing that we wanted to touch on is parameter efficient fine-tuning of our models. So a lot of times there's a consideration to be made between whether you want to exhaustively update all the parameters in a model or just a subset of those parameters. And so there are many different methods of parameter efficient fine tuning, or PEFT as it's abbreviated. As I'm sure many of you are familiar with things like LoRa and DORA, we are opting for more classic PEFT or manual parameter efficient fine tuning, in which we sequentially unlock transformer blocks throughout training so that we are slowly increasing a subset of parameters to update. And so oftentimes, conventional thinking would dictate that if you update all of the parameters, your model is better suited for the downstream task at hand. When we looked into PEFT during both the SFT and PPO phases of training, we see that across model sizes, that actually wasn't true in our hands. We observed that from 8 million all the way up to 650 million, implementing parameter efficient fine tuning at both stages in RLXF leads to no degradation of model performance. And actually, in some cases, as you guys can see in the ESM 650 million regime, you get a slight increase in performance. So you get to balance sort of the pre-training knowledge that you're leveraging with these state-of-the-art protein language models, but you also get to update some of the more important weights that make your model more amenable to the task at hand. And so you can see in this plot on the right with this dark blue line indicating ESM 650, you can see that there is an immense benefit after just one epoch of training using this PEFT method with PPO. And the performance gain that you get, even if you update all the parameters is slightly worse than if you only update a subset of them. So that's the first big thing that we observed as we were doing this. The second big thing is sampling. As I'm sure you guys know, how your model samples residues in a protein that you are trying to optimize is extremely important. And so when we started this, we were simply doing greedy sampling or max sampling. Essentially what that means is that for a given masked residue, the filled in residue is just going to be the residue with the highest probability of existing at that particular position. When we looked into our model performance, though, we were seeing that only the tail end of the distribution of sequences that we output from our models were above the wild type baseline. However, we did a little bit more research, and in the natural language processing field, there is a sampling technique called top P sampling, which in that field had provided immense benefits to model performance. So when we implement top P sampling, what we mean is that we mask every residue of CreiLOV one by one, and we calculate the cumulative non-natural amino acid probability mass for each residue. If that probability mass exceeds a high confidence threshold that we determine as a hyperparameter, we will sample the most probable residue at that position. With this subset of high confidence mutations added to the wild type sequence, we then recompute the per position non-natural probabilities and sample positions proportional to their non-natural probabilities until we reach the target mutation number. In our downstream studies, we are designing 5 mutants and 10 mutants of the CreiLOV protein. And so this sampling method applies to both of those design scenarios. So this allows us to sort of focus down our sampling on functional and structurally important residues while using the model logics to make context-aware mutations. Another advantage of this method is you don't burn your compute masking and refilling in residues that are less functionally important to your downstream fitness. So this is sort of a twofold advantage here in sampling. And you can, on the right, you can immediately see the benefit from switching from simple greedy sampling to a top P sampling scheme, such that at pre-training, the max sampling is better than top P sampling. But then as you move to SFT and PPO, you almost achieve perfect win rates. So we were super encouraged to see this result and implemented this for our RLXF pipeline. The last thing that I wanted to mention as it relates to top P sampling is when we benchmarked RLXF against some of the other popular reinforcement learning methods in the literature, we found that with top P sampling, we were actually able to outperform these methods, even with these methods using P sampling. Now, it is important to mention that the methods that we benchmarked against use an algorithm known as direct preference optimization. Direct preference optimization is a little different in the sense that it explicitly maximizes the log probabilities of a labeled data set of preferred protein variant sequences. So this leads to an extremely sharp learned distribution that seems to benefit from a greedy sampling scheme, which is somewhat intuitive. So even when you use max sampling with these other methods that we've benchmarked against, top P sampling with RLXF outperforms many of these methods. So the last thing that I thought would be important to mention today is essentially, if you are at a point where you're trying to make your model better, you are trying to squeeze all the performance out of your model, the chances are optimizing the underlying training data will make more differences than any of these other sort of micro optimizations. What I mean by that is at first we were doing supervised fine-tuning as a reconstruction task on a subset of the deep mutational scanning data. However, as we were playing around and empirically trying different types of data, we reached a point where we tried using simulated annealing to generate synthetic data for our SFT regime. We generated 4 of these synthetic data sets 1 through 4, as you can see in the light blue box on the right. And when we investigated the model performance with each of these data sets, we found pretty substantial differences in the win rates and predicted fluorescence of each of these data sets. One thing that I do want to mention is that somewhat counterintuitively, the data set that performed the best with our model was data set 1, which only featured 86 of the utopia sequences that were reached using a simulated annealing schedule. 86 training instances doesn't really seem like a lot for a training data set. Generally, the dogma is that more training instances are better, but we found through empirically testing out each of these data sets that really it's a trade-off between quality and quantity. If you have a small number of very high quality training instances, and obviously you've optimized the hyperparameters, etc., this actually can lead to much better performance than a data set that may be 10 or 100 times larger than that. But as you can see in the win rate plot here, data sets 3 and 4, or data sets 2 and 4, which contain several thousand in some instances training instances, were significantly outperformed by the first data set, which only contained 86 simulated annealing synthetic 5 mutant sequences. So I just thought it was important to mention that if you would like to make your model better, oftentimes playing around with the data and even exploring strategies for generating synthetic data of high quality can really push your model to the boundaries of what it's capable of. So with that being said, we arrived at a final model checkpoint using all of the strategies that I mentioned before, and then we were ready to test these designs in the lab to see how well they correlate with the in silico results. So if I just walk you through this plot here really quick, we took our pre-trained VAE and ordered 10 sequences. We ordered 10 sequences from the RLXF-aligned VAE, and all of these 10 sequences contain 5 mutations to the wild-type CreiLOV sequence. We did the same thing for ESM2, where we ordered 10 5 mutants from pre-trained ESM2 and 10 5 mutants from the aligned ESM2. And then based on the mutational extrapolation plots that Nathaniel showed you a while ago, we thought that we should try to order 10 mutant sequences. So we did that. We ordered 10 mutant sequences from pre-trained and 10 mutant sequences from aligned. In addition to that, as baselines, we ordered the wild-type CreiLOV sequence and the T7S single mutant that Nathaniel was telling you about a while ago, which was the best single mutant in the deep mutational scanning data. And what you can see is that comparing pre-trained to aligned models in every single case led to higher hit rates that were greater than wild type. And also, in some cases, with ESM2, you have a couple of these variants, 5 and 10 mutants, that are brighter than the brightest single mutants in the DMS data set. We were super excited to see this result. We believe that some of these, hopefully, maybe some of the brightest flavin-binding fluorescent proteins that have been characterized in the literature, and we have several experiments planned to confirm this. To dive into this data a little bit more, on the left here with the scatter plot, basically this only serves to show you the differences in the mean fluorescence across all experimental groups, where you can see across the board alignment significantly increases the mean for all ESM2 experimental groups. And with the 10 mutants, once again, you can see that even though that mean is slightly under the wild type baseline, we've designed 3 sequences that are brighter than wild type. And this top dot here is approaching 1.7 times wild type fluorescence, which we are very excited about and are certainly going to assay further. The pie charts on the right here, really the main takeaway is just that between pre-trained models and then going through RLXF alignment, you significantly increase the hit rates with only a small number of sequences. Oftentimes in protein engineering papers, you may find that like they will take a model and then order thousands of sequences and they would get a hit. That is not necessary with RLXF. As you can see, we only ordered 10 sequences from each of these groups. And in the aligned ESM2 5 mutant experimental group, you can see that we got a 60% hit rate where high activity is defined as greater than wild type. But even still, with the other 40% of these sequences, moderate activity is defined as 0.7 to 1x or identical wild type activity, and all of those sequences were. Even when you get out to 10 mutations, you still have a hit rate of around 30% for greater than wild type sequences, and in total, 80% of moderate to high activity designs, which is super encouraging, and hopefully it makes this method pretty accessible in terms of the funding that you have available to you in terms of designs, because as I'm sure many of you guys know, it is quite expensive to order so many designs if you're not assembling those libraries by hand. Just the last point that I want to make here, once again, you can see in the sequence logos between pre-trained ESM2 and aligned ESM2, after alignment, you shift the probabilities of preferred amino acids across a subset of residues that are known to be functionally or structurally important. One of the classic examples that we keep coming back to is residue number 7 here, where the pre-trained model almost exclusively puts in a threonine and in some cases puts in a valine. However, after alignment, we entirely put in serines there, which we know to be a beneficial mutation, and this trend extends across many different positions in the protein. And so that 1.7x 10 mutant variance that I introduced earlier is shown here. You can see all of the different mutations that the model put into the wild type sequence. Some of these mutations are very familiar to us, including T7S, G26T, R5D. These are known to be beneficial single mutations in the DMS dataset. And, thankfully, our model was able to pick up on that and substitute them into the wild type sequence. But a bit more impressively, our model also put in some mutations such as P16E that weren't even in the top 100 single mutants in the DMS dataset. And so what that indicates to us is that through this PPO pipeline in which you have a model that generates sequences, it receives a score, it updates its weights, and then tries again, you get to kind of explore this epistatic landscape in ways that we might not be able to do in the wet lab. And so you make combinations of mutations that aren't intuitive but lead to drastic functional enhancements, which we find to be super cool. So just to wrap up, I just wanted to reiterate the fact that we have been talking about an algorithm called RLXF, or reinforcement learning from experimental feedback. And the entire premise of RLXF is to inject pLMs with a notion of biomolecular function, which right now in zero-shot design is oftentimes missing. This enables the generative design of proteins with properties that are tailored to any use case that you may find yourself needing for a protein engineering campaign, given that you have a high quality data set to work with to train a reward model with. This can be anything from binding affinity to thermostability, fluorescence, enzymatic activity, etc. The last quick point that I want to make is that we were able to implement this entire pipeline on one GPU if you're able to or have access to one NVIDIA GPU. And this entire process of training a reward model, generating a synthetic SFT data set, aligning your model with SFT and PPO, and then designing variants. All of this takes only around 2 to 3 days. So hopefully you guys can implement this in your own protein engineering campaigns, and you find that this is easy to work with, it's computationally inexpensive, and hopefully it is pretty powerful. So with that, I just want to acknowledge all of the people that worked on this. This work was led by Nathaniel, and then the undergrads who worked on this are Sarah, Agram, and Amaya, and then myself. Also our PI, Dr. Philip Romero, and then the rest of the lab here for all of their contributions, discussions, and ideas along the way. So with that, we will take any questions that you have, and we thank you for your attention.
Awesome. Let's thank Nathaniel and Shri again for the talk. I will stop the